In this video, I'm going to show you how to perform an Oxford Nanopore genome sequencing run. So this comes with a instructional guide, and it shows you the details of the machine we're going to use, a couple different machines we're going to use. We're going to run the genome sequencer on what's called the MinIT. And what this pamphlet shows you is how to log on and communicate with your cell phone, your computer, with the MinIT device, and how to hook that up to the MinION genome sequencer. So there's instructions on the front with passwords, and there's some instructions on the back of that sheet as well. Here's the MinIT device and how you hook it up. And this is the MinION sequencing device with a Flongle adapter on the inside. And I have two of these. I'm going to run two devices. There's another MinION device with a Flongle adapter to take the Flongle flow cell. And the flow cells inside of these little packets come there. First thing to do is to test the flow cells. And to do this, you go on your cell phone, you go into the Wi-Fi, and you select the MinIT. And the MinIT is called MT-something. This MinIT is MT111256. After you select that, you open up your browser. I have Safari, I guess, on my iPhone. And you type in 10.42.0.1 and click on that, and it will pull up the software that's on the MinIT device. So we're controlling the MinIT and the software with our cell phone right now. You select Flongle flow cell on the right. You click on MN34113, turns it green. So Flongle flow cell. Then you select the MinION device. I have to rotate mine sideways so that I can click on it, my cell phone, and you click check flow cell. And then what you're going to do is the flow cell comes with a code on it. You're going to type in that code. So they give an example there. There it is, AER053. Type that in and start the test. This is going to test that Flongle flow cell to see how many of the pores are active. Now what we want to do is go back, here we go, go back to the Wi-Fi, and I'm going to select the other MinIT device, 111289, which is connected to the other MinION and the other flow cell. Type in the same 10.42.0.1. It opens up the software. Click on flow cell type. We're using a Flongle flow cell. Tap on the name of the MinION. Turn it sideways. Type in the code on the flow cell, AEQ 500, and start the test. If you click on the little envelope there in the upper right, it'll open up that and show you that it's starting the flow cell test. Now while that's going, you want to open up your library kit. This is the Oxford Nanopore library kit, and then I'm going to use the field sequencing kit. You can see it right there. And this it's stable for a month at room temperature, the field sequencing kit. And I've got two of them here because I'm going to run two, you can see them on the left there, two devices, two MinIONs. So you open up the packet, and what you'll see is inside of there are 3 tubes. Here's a silica gel to keep it dehydrated. And what you'll see is there's lyophilized reagents in here. Lyophilized means freeze-dried, and they match the drawing there. Tube 1, Tube 2, and Tube 3. What you're going to do is break off Tube 1. That's where we're going to start. We're going to make our library, starting our library in Tube 1. And there's the second packet, ditching the silica gel. And you see that this also has 3 tubes on it. You bend it and break off tube number 1, and I place those in front of me. Now what I'm showing you are 2 tubes that are full of DNA, L tube and S tube, which contain the DNA from the bacteria that I isolated on the edge of the spinach leaf. So with tube 1, what I like to do is make a little pilot hole there with a sterile pipette tip. In both of these, you can see that, I'm making just a little hole, opening it up. Because what I'm going to do next is transfer in to that tube the DNA. So now I've just showed you tube 2 and tube 3. I've gone ahead and made a little hole in those as well. So it's just easier to insert the pipette tip when I start moving volume, reagents of volume there. Okay, so how much DNA do we put in tube 1? You put 10 microliters into tube 1. So this is the 50 microliter DNA from the extraction. I just take 10 of that, and what you're going to do is pipette that slowly about 5 times into tube 1, mixing the lyophilized reagents with the DNA. So ditch the tip, got a new tube, a new tip, pipette tip, 10 microliters of DNA from DNA sample number 2, and hard to see because it's blurry, but pipetting up and down about 5 times, mixing the DNA, there we go, mixing that DNA into the reagents in the bottom of that tube. Now this reaction is supposed to take 1 minute, and you can do this at room temperature, perform this at room temperature, or you can put it at 30 degrees Celsius. I put it in at 30 degrees Celsius for about a minute, and you can see it's already counting down. So while that is going, I'm going to go ahead and set up the other tubes. So what you see here is a resuspension buffer, and 65 microliters of that is going to be pipetted into tube 3 for each of those that are in front of me. So 65 microliters into that. It's really a loading buffer that's going in there into tube 3. So what's happening in that PCR machine, I use it as an incubator, and you're going to pipette up and down on this, by the way, until it mixes. What's happening in that PCR machine is that it's going to go 1 minute at 30 degrees Celsius, and it's going to go 1 minute at 80 degrees Celsius. And what's happening during those processes, or those temperatures, are there is the DNA is being cut in the first step, and an adapter is being attached. The second step that's going to happen next is we're going to attach another adapter to that tube after we take it out of the PCR machine. And that second adapter is a sequencing adapter. Okay, so what you just saw there, so that's running off to the side while I'm doing this, is we've added the resuspension buffer 65 microliters to tube 3. I also have another tube right there in front of me in the rack that has the lip towards me, or the cap, and I'm now setting this up here at 6 microliters, and I'm showing you 6 microliters, a flush tether. So we're going to prepare the flush, what they call the flush buffer. We're going to prepare the flush buffer times 2 into that tube in front of me. So I'm taking 6 microliters out of that flush tether and putting it into the tube. So we're just doing 2 things at once here. Normally what you do is 3 microliters of the flush tether, followed by 117 microliters of the flush buffer. But because I'm doing times 2 here we go, we have 6 microliters and 234 microliters, and mixing those together. So there it is, the flush buffer. So normally 117 and the other 3. We're doing 234 and 6 to double it up because we have 2 samples, 2 MinIT sequencing devices. So everything gets doubled in this case. So you can see I'm mixing in that 234 into that 6, which makes 240. So the flush buffer is ready. These samples are done because they've done 1 minute at 30 degrees and they've done 1 minute at 80 degrees And now, turning that off and trying not to burn myself. Put those back in front of me. Keep them separated. They don't need to be labeled because I know the one on the left is L, the one on the right is S. So this is the P10. I put it at about 10 microliters. I put it a little higher because the lyophilized reagent plus the liquid is actually a little higher than 10 microliters. And we're going to take that out of tube 1 after that incubation and pipette it down into tube 2. Here we go. So pipetting up and down just like before, 4 or 5 times, somewhere around there, kind of slowly and kind of gently into that tube. So from 1 into tube 2. Here's the other sample, which you can actually see. Take out that 10 point whatever. Take out all of it. Put it into tube 2. And there you go, you can see that. Pipette up and down 4 or 5 times smoothly. Try and be gentle with that until that's done. That's going to incubate for 5 minutes. So while that incubates for 5 minutes, then what you do is you go back to your MinIT devices, and it shows that one of these has 65 pores that are available for sequencing. So that's great. That's what you want. The company Oxford Nanopore guarantees they have at least 60 pores available. Then what you see me doing here now is going in and setting up a sequencing run. So I'm typing in new experiment. You type in the ID of the flow cell, then you give it a sample name. Then at the top you need to give it an experiment name, whatever you want to call it. It has to be different than the sample name. So you got an experiment, a flow cell ID, then you have to select a kit, which is LRK001 for the field sequencing kit. So that MinIT sequencer with its flow cell is all ready to go. All right, so what you're seeing here, so I just showed you one of those. I didn't show you the other MinIT device. What you're seeing here is instruction from the company on how to do the next step. Because what needs to happen next is the Flongle flow cell needs to be placed into the device like you see here. The tape needs to be pulled off and the flush buffer needs to be added, 120 microliters of it. So you're seeing the tape being pulled off of that, the instructions, pushed against the lid, and then below you're seeing that it says don't introduce a bubble. So you're going to add 120 microliters of that 240 that we mixed, it's called the flush buffer, into the pore that's in the center there, without introducing a bubble, which would destroy the 65 pores that are active, or at least that it tells us. After that, you're going to pipette in 30 microliters of your sample. Here's a video on how to do that. is one of the Flongle flow cells, and I'm doing what you saw in the video there. I'm pulling back the tape. This is the flow cell AEQ 500. You see on the left there's that little code you have to enter a couple times when you're checking the flow cell, but then also when you're setting the experiment up. You push the tape into the lid of the MinIT device. You get your 120 microliters now into your P200 pipette, and you want to put that down into the pore and slowly, evenly pipette that onto or into that pore without introducing a bubble. It's kind of hard to see here what's happening. I've done 24 of these, and I can tell you that it's not easy. So it takes a little bit of a technique. I'm going to show you on the next video. This is me failing. So these fungal flow cells cost 90 bucks. The field sequencing kit that I just set up, that costs $100. So you've already set that up. You're trying to pipette in. Now you're flush buffer so you can load your sample. And when this happens, if the liquid does not go in, it's actually coming back out and forming a large dome on top of that little concave pore-like structure. When that happens, that gets frustrating because the device is ready to run, the sample's ready to run. We set that up. We know it has some pores, but you can see that I'm struggling to get the liquid to actually go into the sample port. I keep calling it a pore, but it's a sample port. So eventually I got that one in. This is another one, which I ran the next day, showing you with a little bit better video how it works. You want it to work. And I think the technique here is there's a little sample port. The technique is to slightly put pressure on it, but to almost point the pipette tip back towards you. And you can kind of see that in that other video that Oxford Nanopore produced, the previous one, but they don't really tell you that. So here it is with the tip going in. You're going to see me kind of put some pressure on it and bend out away from me, and I'm kind of pointing it towards me. And you'll notice there's a little bit of fluid actually moving through the outside of that. You can see it moving there. So this one went in. The other two that I was running for that, the day before I ran this one, it took me about 5 minutes to actually get those in. And I actually lost a lot of pores. I destroyed them because some bubble went in there. And I pipetted back and forth a few times, which you don't want to do. The goal is to put that into there and dispense it or pipette it all in one shot smoothly like you just saw. So what's happening now, you don't see me. I'm putting a new pipette tip on the pipetter, and I'm pulling up 30 microliters of the library, which is, by the way, a mixture of tube 2 and tube 3. So when you mix the 10 microliters from tube 2 into tube 3, which had 65 microliters of a resuspension buffer, you have 75 microliters. For a Flongle, you only need 30 of that. So there it went. The sample went in fine. You gently place the tape back on there. You fold it back down over. There's a few other ports on there. There's a waste port. Cover those up with the dots that you see there and close this up. Then you go back to your run and click on Start Run because you've already set it up. So there it is. This is the experiment lettuce number 2, and that's going to start. If you click on the little envelope in the upper right, you'll see that there's it's going to warm up to 35 degrees Celsius, and then it's going to start the run. So right now we've gone back to the other experiment. It says lettuce L2. This is all on my cell phone here. And this one is firing up. It's almost there. It's working its way up to 35 degrees before it actually starts. Now I'm going back to my Wi-Fi, it looks like. And I'm changing to the other MinIT device. I have two different numbers, so I'm going back to the MinIT 111256. Then I have to refresh all this because I had actually gone to the other device. You can only communicate with one device at a time. This one says it had 57 pores available for sequencing. And so what's happening here now is I've selected a new experiment to run the second device. So what's going to go on here is I'm typing in the flow cell code. AER053 was the code on that other flow cell. So it looks like these might be a tiny bit out of order how I've structured them here. But it's the same process. You check the flow cell, and while you're checking it, then you go ahead and set up your experiment. You select your kit, LRK001, which is the field sequencing kit. And then you wait until you load the sample. After you load the sample, then you'll click Start Run. So now I've gone back into the other MinIT. Confusing, isn't it? And I'm refreshing because the software needs to be restarted. I'm reestablishing communication with the software on that MinIT device. Now the experiment's actually running for LetSL2. It tells me that I lost a lot of pores, which is frustrating because I had trouble loading it. There's only 35 pore sequencing out of 126. So very frustrating, but that's kind of how it goes. It's challenging sometimes to get the buffer in there, then also your sample. Now what you're looking at is called the channels path in the actual live sequencing that was recorded right from my cell phone. And what you're seeing are some pores that are sequencing. Those are the light green ones. Some pores that can sequence, those are dark green. They're available for sequencing. You've got some that are recovering, and you've got some that are inactive. So now I'm scrolling down. The inactive ones are the light blue, and they've got histograms of these. So you see them there. Light blue means it's inactive. That means it's destroyed by some sort of means. And then down here, this is the cool part. This is read length histogram. So already in these first couple minutes, the first 20 reads, if you look at the top, we've got some reads that are, it looks like one that's about 10,000 nucleotides or bases long, one that's about 5,000 something, one that's about 3,000 something, and some others that are in the 2,000 range and so forth. So we have some sequencing reads that have already been base called, they've already been analyzed, and they're sitting right there on that device. So at this stage, now you can see at the top it says 2 hours and 8 minutes. This has been fast forwarded. There's 793 reads. Estimated bases, 922,000 bases or nucleotides. We've got less pores that are actually sequencing. Most of them are inactive at this state, but we still have, looks like 95 minus 126. So we still got some pores that are sequencing after a couple hours, which is fine. And there it is, kind of the breakdown of that. But you can see we've got a lot more now. We've got a 15,001, looks like almost a 20,000 off to the right. I've magnified it for you to see a little here. A bunch of 10,000 nucleotide long reads, and that's awesome. So we've got 795,000 reads after a couple hours there.